I'm Rachel Hostetler. I've been running this webinar series called Cell Type Taxonomies A to Z. And today I am joined by Kelly Jin, who is the lead author on our new transcriptomic cell type aging study. And she focuses on mouse cell types in the brain. And now we'll begin. And as a reminder, this webinar is being recorded. It'll be up on our YouTube channel afterwards at Allen Institute. And this webinar is part of a webinar series. So we'll be having a number of events as well in May and June that you should sign up for if you're interested in some of our future topics. And we are also happy to take questions during this webinar. So there's a Q&A function on Zoom that you can enter your questions in, and I will be moderating those questions. And we'll go through those questions at the very end of the talk. And if you're watching a recording of this webinar on YouTube and you have questions, please visit our community forum, which is linked in our YouTube description. It's also community.brain-map.org. That's our online community forum, where we have many scientists from Allen Institute moderating the forum and answering questions. And so if you have any further questions, please post them there. And as a reminder, we are from the Allen Institute, and the Allen Institute is a nonprofit bioscience research center in Seattle. And we have three main principles: team science, big science, and open science. And so team science is where we have multiple large interdisciplinary teams working together to tackle very large projects, such as the aging mouse brain. And so when you see a paper from the Allen Institute, there's a reason why there's so many author names on there. We have a lot of different people from different teams doing different aspects of our experimental pipelines to create these very large data sets. And so we also focus on big science, creating these very large projects with robust data. And then we share all of these data sets and projects online openly for free on our website, brain-map.org. And in the second half of this webinar today, I'll be featuring some of our tools and other resources that are related to the aging mouse brain dataset. And now I'm going to hand it off to Kelly.
Thank you, Rachel. Let me get started and sharing here. So good morning, everyone. Thanks for joining us. So everything I'm going to talk about today is in this paper that we published earlier this year in Nature. So if you are curious about any additional figures or data that I'm going to talk about today, please go look in this paper. This will also include all of the information about how you can access the data and the different tools that Rachel is going to talk about later a little bit more. And first, I just want to acknowledge not everyone, unfortunately, because I can't fit all the names here, but a lot of the key people who played a big role in helping collect, analyze, and interpret this data throughout the whole process. Okay, so before I start talking about aging at all, I wanted to mention something, another resource that plays a big role in this data set and in a lot of data sets that moving forward that we'll be collecting and talking about at the Allen Institute. And that's this resource called the Whole Mouse Brain Taxonomy, or some people might call it the ABC Atlas or the ABC Whole Mouse Brain Atlas. And this refers to this really amazing resource paper that was published a little over a year ago by some of my colleagues, also in Nature, and here's the reference. This data set represents a high-resolution transcriptomic and spatial atlas of what we think is close to all cell types in the whole mouse brain. And so this is a gigantic data set of a little over 4 million cells, single cells from the whole mouse brain that was broken up into about 50 different regions. Each of those different regions were separately processed into single-cell RNA-seq libraries. And what you see here is the UMAP representation of all of those cells. And my colleagues built a very detailed high-resolution taxonomy from all of these cell types. which then they mapped onto this spatial data collected with VisGen's MERFISH platform to visualize where all of these different cell types physically may be in the coronal mouse brain space. So just to get into a little bit more detail about the different kind of nomenclature that we use for all of the taxonomies associated with mouse brain, the kind of the broadest cell category that we use to talk about cells, we call class. And I know these labels are a bit small, but you can see that most of these different cell classes are grouped um, by either glutamatergic or GABAergic neuron cell types. But we also have our major glial and non-neuronal cell types down here. Within class, we have different subclasses. Within subclasses, we can further break that down into what we call supertypes. And then finally, within supertypes, there are individual clusters. And so in total, this massive taxonomy represents a little over 5,300 transcriptionally distinct clusters, most of them being neuronal cell types, and a little over 110 clusters representing the non-neuronal and glial types. Okay, so as I've alluded to historically, we at the Allen Institute have been building tools revolving around the adult mouse brain. So trying to annotate, characterize, and identify all of the cell types in a healthy adult mouse brain. So kind of creating this parts list of all of the potential parts or cell types that make up a brain. Now that we've released this amazing resource, this giant taxonomy, we can start to begin to bridge these different types to function and try to better understand healthy brains, for example, at different ages or different disease, and which parts or which types, cell types, might be the biggest players in these different in aging or disease, and of course, how these types or these cells start to breakdown, which may ultimately lead to disease. And aging, of course, is the major risk factor for just about all leading causes of chronic death, leading causes of death in the developed world. And so the idea is if we can better understand aging, perhaps we can not only better treat these diseases, but hopefully better prevent their onset as well. So all of the data I'm going to talk about today come from 2-month and 18-month-old animals. And in the rest of the talk, I'll refer to these two ages as adult and aged tissue. So this is a large single-cell RNA-seq data set collected with 10x Genomics version 3 technology. We profiled these 16 specific regions of the brain, so not quite the whole brain, but we cover these six broad brain regions. And for the rest of the talk, I'm going to mostly be grouping these samples by these 6 broad regions and associating with the same colors as well. So just for simplicity. In addition to this large single-cell RNA-seq data, we also collected some in situ spatial RNA profiles using a platform from a company called Resolve Biosciences. So this is very similar to kind of the MERFISH technology that I showed earlier. But this particular technology offers a bit more targeted profiling, so smaller brain regions and a smaller gene panel. So it's meant for more kind of hypothesis-driven validation of gene expression changes that were identified from the single-cell data. So to talk a little bit more about how this data set was collected, these fine regions were dissected out by our very talented mouse teams, and about half of the samples were processed using an unbiased sampling method, and the other half were sorted for SNAP25 positive cells with the intention of enriching for kind of more neurons in the data set. Downstream 10x genomics and Illumina sequencing workflows were followed. And finally, label transfer was performed using labels from the ABC Whole Mouse Brain Atlas that I described earlier. Um, so this is just kind of a summary of the different um, levels of labeling in this data set. Again, you can see that we have class, subclass, supertype, and finally clusters. And in this data set, there are a little over 800 clusters. And just to give an example of kind of the breakdown of this structure in the astroepinemal class, we can see, within this class, there are different subclasses, which include different kinds of astrocytes, tanocytes, epinemal cells. Within one of these astrocyte subclasses, there are different supertypes. So TE stands for telencephalic, and T stands for non-telencephalic. Within one of these supertypes, then we can break this down even further into transcriptionally distinctive clusters. Um, and so this is just a UMAP representation of all of the cells in the data set. So here, this UMAP is colored by cell class. So you can see about half of the cells are coming from non-neuronal types, and the other half are made up of different neurons. And if we compare this with the same UMAP that's colored by major brain region, as we would expect, you can see that the glia and the non-neuronal cell types are kind of scattered throughout all of the different six major brain regions, whereas the neurons are a bit more localized to specific major brain regions. And finally, we can color this same UMAP by age. And at this level, I think this more just kind of tells us the different sampling biases of this data set. There's, some regions have slightly more adult cells, some regions have slightly more age cells. But in total, this data set is a little over 1.1 million high quality single cells, single cell RNA profiles, and a little over half of those come from aged animals. Okay, so now that we have these really fine resolution labels, we can approach, um, asking these biological questions in a couple of different ways. So firstly, the first thing I did was looked for age-associated differentially expressed genes, which I'll refer to as age DE genes for the rest of the talk at these different levels, so subclass, supertype, or cluster levels. We can also ask if there are any clusters, so the finest the finest level of cell types, if there are any age biased clusters, so clusters that only show up in the adult samples or clusters that only show up in the aged samples. So just to summarize the entire data set in terms of for HDE genes, this plot is showing for every column is a unique cell type, and this is broken up at the level of non-neuronal supertypes and neuronal subclasses. And these bottom three bars are kind of just summarizing the major metadata categories. So similar to what we saw from the UMAPs, we can see that our non-neuronal types are coming from all kind of major brain regions, whereas the neurons are specific to specific regions. And we can see the breakdown of each of these classes or each of these subclasses and supertypes by age and sex. And finally, this topmost plot here shows the total number of HDE genes that we found across each of these different cell types. And what might draw your attention right away is that we see a lot of changes happening in these non-neuronal supertypes as compared to the neurons. And in total, if we look at all of the different genes that this represents a little under 2,500 unique age-associated differentially expressed genes. Furthermore, if we take all of these genes, all of these about 2,400 genes, and make a histogram of the number of types for which a gene is significant, so just to be clear, this number 80 means that these genes that I've listed here are significantly changing with age in about 80 different cell types. this is what the distribution looks like. And we see a handful of genes that seem to be changing in quite a few different cell types, and we refer to them in the paper as common aging genes. But you'll see that the vast majority of those 2,400 genes that we found were changing in just one or two specific cell types, which suggests to me that most of these genes are cell type specific, and all of these cells are changing in pretty different ways with age, which makes the single cell approach to looking at brain aging very, very important. Next, I'll zoom in a little bit more on these non-neuronal cell types, since that's where we saw a lot of gene expression changes. So this figure is essentially the same figure I just showed in the last slide, but kind of flipped on its side. And now with the different non-neuronal supertype labels shown. So we have our astroepinemal cells, different types here. We have our oligodendrocyte types, vascular cells, immune cells, and I've also included immature neuron types here as well. So this is again summarizing the total number of age DE genes we saw across at the supertype level across all of these different cell types. This panel now kind of dives into the cluster level of the taxonomy. So again, each row represents a single supertype, and each point in this in panel B now represents a single cluster that belongs to each of those supertypes. And what's plotted on the x-axis here is a log2 odds ratio of the number of aged cells within each of these clusters. So what this represents, what a negative log2 odds ratio would represent are clusters that have fewer aged cells than we would expect by chance. And then vice versa, a more positive log2 odds ratio represents clusters with more aged cells than we would expect by chance. And so the way that we decided to call these different clusters are clusters, that below or above a certain threshold as being depleted with age. So these are, since they're depleted, that means they're enriched in the adult samples, or clusters that are enriched with age. And so one thing that we can notice if we look at the cell types that seem to be depleted with age, as well as the cell types that have the largest numbers of HDE gene counts. A lot of these types that are highlighted here are cell types that seem to be associated with regions of known or suspected adult neurogenesis. So that includes types that localize to the subventricular zone near the striatum, which includes a specific telencephalic astrocyte type that localize closely with these immature neurons in the subventricular zone, the subgranular zone in the dentate gyrus. Again, there's an astrocyte-like cell type that localizes very closely with the immature DG neurons. And then finally, a lesser known area of suspected neurogenesis is the third ventricle of the hypothalamus, specifically these tanocytes and ependymal cells that line this ventricular area, which I'll talk a little bit more about later. Another set of cells that I wanted to highlight here are these OPC oligodendrocyte, this group of cells. So I've typed out here what these acronyms stand for. So we have our OPCs, which are the precursors. COP stands for committed OPCs, and then newly forming oligos, myelin-forming oligos, and finally mature oligodendrocytes. And this order is the same order which they appear to be maturing along, at least in this UMAP space. So we have our OPC island and these different transitionary types leading to the more mature oligodendrocytes. And you'll notice here that there are quite a few age-biased clusters that we see, including these newly forming and myelin-forming oligodendrocytes, which seem to be depleted with age. So that's to say that we mostly only see them in the adult samples and a couple of mature oligoclusters, which we only see in the age samples. And that can also be inferred by just looking at the UMAP. We can see this kind of bridge of transitioning cell types is much more green, suggesting that perhaps this transition between from OPC to mature oligos is either not as efficient or just starting to turn off completely in aged animals. Okay, so the next cell type within these non-neuronal cell types that I wanted to talk about are these tanocytes and ependymal cells, which um, some people might not know about. Had never heard of a tanocyte before I began this study. So just for anybody who's not too familiar with these cell types, ependymal cells are these kind of small cuboid glial cells that line all of the ventricles in the brain and the central canal of the spinal cord. They assist in the circulation of CSF throughout the ventricular system. Tanocytes are a very specialized ependymal-like cell, but they are only found in the ventral portion of the third ventricle of the hypothalamus, which I've diagrammed here. Each tanocyte has a single long process that extends into the parenchyma of the hypothalamus, and it interacts specifically with blood vessels that are found in CVOs or circumventricular organs of the brain, which, we can have a whole nother seminar about CVOs, but these are really interesting areas of the brain that actually lack a traditional blood-brain barrier. Um, so, uh, so, you can imagine one end of the tanocyte is facing your CSF, and the other end of the tanocyte is facing these leaky blood vessels. So they are a really critical cell in that they control the dialogue between stuff that's in your blood and stuff that's in your CSF. And they help kind of modulate how leaky these CVOs or circumventricular organs are. So you can imagine they're really important in sensing different kinds of nutrients, sensing different kinds of hormones, maintaining energy homeostasis. And another crazy thing about them is that they're also, many people suspect them to be neurogenic and gliogenic into adulthood. So they're really amazing cell types. And of course, we saw a lot of genes changing with age, and specifically in these tanocytes. And so here I'm just showing a heat map of a handful of those genes, not all of them, along with ependymal cells and the two major astrocyte types. And you can see that these gene expression changes are very specific to tanocytes and to ependymal cells. And some of them are decreasing with age, some of them are increasing with age. And indeed, we were able to use our in situ resolve spatial data sets to confirm that. So these 4 genes are, I've also pointed to here in the heat map, and we can see that they are increasing or decreasing in the way that we would expect across both ependymal cells and tanocytes. Furthermore, if we also performed gene ontology enrichment analysis in these genes that were changing with age, and just see a variety of really interesting biological processes that seem to be going on in these genes that are changing with age. So a darker bar represents genes enriched in genes that are decreasing with age, processes that are have enriched in genes decreasing with age. And then the lighter gray bars are genes increasing with age. And then these red bars are terms that are unique specifically to these 2 subclasses. So they're terms that aren't seen elsewhere. So for example, on tanocytes, we see a decrease in a lot of genes related to neurogenesis, neuron differentiation, which may have something to do with their neurogenic capacity. We see terms like dynein complex, amyloid beta binding, and these terms are unique specifically to tanocytes. And then for ependymal cells, we see a huge upregulation in MHC class 1 protein complex, interferon-mediated signaling, So there's very strong gene expression signatures coming from these two relatively rare cell types. Okay, so I've talked a lot about glia. I wanted to say something about neurons. So we also see a decent number of changes happening in different hypothalamic neuronal types, which I'm going to highlight here. So if we look at the top six subclasses, hypothalamic neuronal subclasses with the greatest numbers of HDE genes, and if this is a snapshot again from the ABC Whole Mouse Brain Atlas, which Rachel will feature a little bit more later, you can see that these types, these neuronal types, are specifically localized to regions that surround the third ventricle, like the paraventricular hypothalamus, the dorsal medial hypothalamus, and the arcuate nucleus. If we dive a little bit deeper into the clusters that make up these subclasses, and look at specific marker genes that are associated with these clusters, we see something pretty interesting. So a lot of these markers might sound familiar to you, like GLP-1R or leptin receptor, AGRP, NPY, POMC. These are all neuropeptides or receptors that are highly are very famous for kind of their role in energy homeostasis and regulating hunger and satiety response. And it's pretty interesting that a lot of these hypothalamic neurons with the greatest changes, gene expression changes with age, are known for kind of their role in kind of controlling energy homeostasis. Okay, so now to try to kind of tie different cell types together, I performed gene ontology enrichment analysis across all cell types. And I wanted to try and summarize kind of commonalities, common terms, enrichment terms that we saw across different cell types. So this is a giant matrix heat map, and each row is a different cell type, both neuronal types, which are colored with by the major brain region they come from, as well as non-neuronal types in gray. And then each column, which of course have had all the labels removed, is a unique gene ontology term. And so we can see that a lot, and this is color. The color that they represent is kind of a geoterm significance score. And so the scores that are more positive or colored red are terms associated with genes that are increasing with age. And then blue are terms associated with genes that are decreasing with age. So we can see a lot of these blue terms that are popping up are mostly associated with neurons, which might be a little bit hard to infer from here. And these terms are largely have to do with neurogenesis, neuron development, neuronal structure. And then a lot of these red terms here we see enriched in non-neuronal cell types, which have to a lot, and many of these terms have to do with inflammation, immune response, cell migration, and motility. And just to zoom in on, a very small portion of this matrix, because there's obviously a lot of information here, I wanted to highlight kind of some of the one finding I thought was particularly interesting, which is enrichment of these terms related to MHC1 and MHC2 response. So these are your major histocompatibility complexes, which have a lot to do with antigen presentation and recruitment of immune cells. So it's perhaps not surprising to see these terms enriched in microglia and BAMs, which stands for border-associated macrophages, since they're immune cells. But I was pretty surprised to see such strong enrichment of these terms in other cell types, other non-immune cell types, like tanocytes, ependymal cells, endothelial cells. And only in one very specific neuronal type, and this is a hypothalamic neuronal type, specifically one that expresses leptin receptor and GLP-1 receptor, we also saw very strong enrichment, even stronger than in the microglia. And you can see we can further break down these terms by the specific histocompatibility genes that drive these terms. And you can see that there's a very distinctive difference between MHC1 and MHC2 classes. So just to summarize, kind of major trends that we saw across the entire data set, we saw mostly in our glia cells, a lot of increase in genes related to immune response and inflammation, not only in our immune cells, but also in some of those other non-neuronal cell types like vascular, like endothelial cells and tanocytes. And then across our neurons, we saw a lot of mostly decrease in genes related to neurogenesis, neuronal signaling, and neuron structure and integrity. And perhaps most surprisingly to me, that came out of this study was a lot of these types, these cell types that surround the third ventricle of the hypothalamus, which have to do, which include a lot of cells that are very well known for their role in nutrient sensing and energy homeostasis, are showing both of these kinds of changes, both an increase in immune response and a decrease kind of in neuronal structure. And these include cell types like ependymal cells, tanocytes, and those different energy homeostasis neurons that I had mentioned. And if we kind of try to tie this to what others in the field have been seeing, this may make a lot of sense. So here's a nice figure from this review paper, kind of laying out different hallmarks of brain aging. So we see a lot of evidence of different kind of hallmarks that they lay out here, including glial cell activation, stem cell exhaustion, which we saw in the depletion of these, a lot of these different kinds of immature neuron associated populations, aberrant neuronal network activity, and the decrease of these genes related to neuronal signaling and structure. I mean, of course, the big hallmark that underlies all of these is dysregulated energy metabolism. Um, so, so we're really excited by these findings, and we hope that the community is excited about using this data set to help study whatever kinds of specific questions about aging that you guys are interested in. And that's all I have for today. So thank you for attending. And next, I'll turn it back to Rachel to kind of give you a demo of how to use some of our tools associated with this data set.
Yeah, thank you, Kelly. I also noticed that we did have one question related to your portion of the talk, so I figured we should probably do that now before I jump into my part. The question is, were there cells located spatially near those cells with upregulation of related processes, like an inflammatory glial cell that might have been secreting something similar that can set off ependymal cells?
We we weren't able to spatially profile large regions like in the same way that the ABC that the whole mouse brain taxonomy did. There were smaller regions. So I didn't see specific cell types localized around. Um, I'm assuming maybe you're talking about like immune cells. Um, but that's a very interesting question that perhaps you can also use our data set to look at.
Yeah, I think for if inflammatory glial cells that might have been secreting, yeah, interferons or other something similar was the question. Um, yeah, okay. We can move on to the next.
Well, let me let me backtrack. We did see localization of specific macrophages to to a specific tanocyte cluster with age. That was actually one of the figures that I took out last minute because I didn't want to go over time. But it's in the paper, so you can also look at the paper specifically if that was your question.
Yes, many figures and also extended figures and supplemental data that's associated with the paper. So please, yes, check that out if you have further questions. All right, so now I'm going to jump into showing how you can access and use this data set using a couple of our different tools. All right. So I'll mainly be focusing on the ABC Atlas, which stands for Allen Brain Cell Atlas, and you can access the ABC Atlas either by searching just ABC Atlas or going to brain-map.org, atlases and data /BKP/ABC-atlas, or we have this QR code here as well. Or if you go to our main website as well, brain-map.org, we also have multiple links to get to the ABC Atlas. But here is the main landing page that we have for this tool, and there's at least 4 or 5 different data sets that you'll find on the landing page, and you'll have to scroll down a bit to get to the cellular and molecular characterization of the aged mouse brain. And that is the data set that Kelly just presented. And then here's just a short description talking about this data set. And right here in gray, where it says brain-wide cell type specific transcriptomic signatures, this is actually a hyperlink to the article that's published in Nature. So this is another way of accessing that article is by clicking on this hyperlink. And then to actually access the data set in the Allen Brain Cell Atlas, we have this nice big blue button here. And then later I'll show you what these green buttons do as well. But if we click on the blue button, this is the landing page for the, or this is the main entry page for this data set here. Oh, just a moment, I lost my mouse. Okay, let's see. And then, okay, there we go. So this is the main UMAP that is shown when you click on this aging data set here. And it's listed up here, 10x single-cell RNA-seq, aged adult, 18-month and 2-month. This is the UMAP here. If you've used the Allen Brain Cell Atlas before, you'll see that it's laid out in the same way as a lot of our other data sets. There are a few extra filters here that are specific to the aging data set, such as the cluster age bias and the log to cluster odds ratio. But as Kelly mentioned, this data set was really integrated with the whole mouse brain taxonomy. And I'll show that here in this video. So this video will demonstrate how to look at the different classes within this data set. So the classes are color coded. The pink is our ITET. We have our other colors as well. And these colors are the same as in the whole mouse brain. So right now I'm showing how you would open up the whole mouse brain data set side by side. So if you click on 10x whole brain, you can have 2 separate data sets present at the same time in the atlas. Here I'm just zooming out so you can see the UMAPs better. And because these taxonomies, the aged brain was mapped onto the whole mouse brain taxonomy, if you see a class or a color in one, it's the same in both data sets. So here we have, if you highlight one area of the UMAP, it'll show up and be highlighted on the other UMAP, again, because these are linked at the class, subclass, and supertype levels of the taxonomy. At the cluster level, things are different because the aged clusters were clustered differently compared to the whole mouse brain. But for class, subclass, and supertype, it's the same. So here I'm showing supertype, and if you hover over one supertype on the UMAP, you'll see it also highlighted in the corresponding whole mouse brain as well. And on the side panel there, right now it's listing the different classes, but then you can see those color-coded bars. And since I currently have the UMAPs colored by supertypes, these color-coded bars are showing the proportions of those supertypes within each class. And so this is just showing how you can jump between the aged mouse brain taxonomy and the whole mouse brain taxonomy at those different levels. Okay, and as was previously mentioned, that whole mouse brain taxonomy also includes MERFISH data. And so Kelly mentioned this figure earlier, where we had these astrocytes and these immature neurons that were both aging depleted, specifically in an area known for adult neurogenesis within the subgranular zone here. So that's what I'm going to highlight in this next video, is showing how you can actually recreate this figure and find those cell types in the whole mouse brain Merfish data set using the ABC Atlas. So first, what I'm going to do is exit out of my UMAP. I click the trash can because now I need to add in the Merfish data set. So if I go to add visualization, we have a couple different Merfish data sets in the system, and that just depends on whether or not you want extra imputed genes or not, or if you want the reconstructed coordinates, which is the common coordinate framework that outlines the different anatomical areas of the Merfish data. But now I'm specifically going and I'm looking for that astrocyte supertype and that immature neuron supertype that's depleted in the subgranular zone. So I'm typing in my different cells of interest. I filter them in the aged UMAP on the left here. And now I'm going to zoom in on one of the Merfish sections. So now we can see those corresponding supertypes in the Merfish data as well. And so if I go and hover over the red cells, the red astrocytes, you can see them pop up in the Merfish data. And same with the brown immature neurons. If I hover over them, you can see them pop up in the Merfish data. It's a little difficult to see with all the supertypes, but now I'll filter it so that it's just showing those astrocytes and just showing those brown colored immature neurons here. There we go. And so again, even though these are different data sets, this is the age data set on the left and the whole mouse brain adult Merfish data set on the right, because they were linked through class, subclass, and supertype, you can use these data sets together to look at the cell types in both data sets. Okay, and now if we want to just focus on the aging mouse data set, so we won't look at the whole mouse brain taxonomy anymore, here's an example of how you can look at specifically looking at age bias clusters within this single cell data set. So you can actually have two of the UMAPs side by side, so I just doubled it by clicking on that plus button, and now I'm actually going to sync these two views together so that when I zoom in and out or move one screen around, that they're synced. And so sometimes this is helpful if you want to have two UMAPs side by side, and then you'll have two different filters or two different labels on those two UMAPs. So on the right, I'm coloring by age. I'm having it colored by age biased clusters. And so green represents aging depleted clusters, and brown represents aging enriched. And if I have it side by side with my other UMAP that's showing class, I can see that both the aging and enriched are in the same class. I just switched it to subclass, and so both of these are also in the same subclass. But once I get down to supertype level, I can start to see that now we have differences between our aging enriched and our aging depleted groups. And then if I go all the way down into cluster level, that's where I can really see that if I zoom in here on this aging depleted group, here in green, that there's at least 5 different clusters that are within that aging depleted group. And if I hover over one, you can see the dots, the Merfish markers pop up on the other side. So now I'm looking at the aging enriched, and I can see that that light green is part of my aging enriched group. And if I zoom in more, I can see that this other brown cluster here, this dark brown, is also within my aging enriched cluster here. And I can also color code these filters by this UMAP by multiple filters at the same time. So now I'm looking at clusters that are depleted or enriched. And another question might be, well, where are these located in the brain? So I just selected the aging enriched clusters, and now I'm actually going to color those same cells by anatomical division. And then my isocortex is in green, my hippocampus, my striatum's in blue. And if I go all the way down to the midbrain and pons, that's where we really see the majority of cells pop out. And you'll see that in the left-hand panel there, we have these numbers. And these numbers actually correspond to the number of cells that are in those different anatomical divisions based on my filter. So right now I have the entire data set, which is about 1.16 million cells, and I filtered it down just to aging enriched clusters because I have that check mark down here in the lower left-hand corner. And then if I just select those, now I only have 3,000, about 4,000 cells, which you can see in the upper left-hand corner. And then within those roughly 4,000 cells, I can see that about half of them are located in the pons. So these numbers in the side panel here update depending on which filters you have selected. Okay, so now if we want to look at specifically looking at differentially expressed genes associated with aging in our OPC oligo supertypes, here's how we would do that. So I went and selected these 4 particular supertypes. We were just looking at this area of the UMAP in the previous slide. Here we have 4 different color-coded supertypes based on these ones selected here. And so with the ABC Atlas, if you know which genes you're interested in, you can go and click on this gene symbol here and search individual genes. But if you have the opposite question, you're looking at a supertype and your question is, well, which genes are differentially expressed in these supertypes? Then we actually have to move to another resource to answer that question. And so the next resource is our Jupyter notebooks. So if we go back to that main landing page here, again, this is our aging data set, cellular and molecular characterization of the aged mouse brain. We have these 2 green buttons for programmatic access and Jupyter notebooks. These take you to the same GitHub page. And if you click on that link, this is what this looks like. We have a very straightforward, easy to use notebooks that have been made by members of our data and technology team here. They're all listed here on the left. And so since we're looking at the aging mouse notebooks, that's the one we want to click on. And then I specifically clicked on mouse aging 10x RNA-seq gene expression. And then so using these notebooks, what I did was I scrolled down to the part that says top N age DE genes. And this top cell here is all copied over from that notebook. Down here is also copied over. This is from the notebook as well. And you can decide, you can edit how many top genes you want to select. I said I wanted the top 2 differentially expressed genes from each supertype. So I edited the number 2 in here. And then I also added in this cell down here, this section, because the way that the notebook is set up right now, it'll give you all of the top genes for all of the supertypes or subclasses or cell types. And I specifically wanted to filter down to just these 4 supertypes here, the OPC, COP, MFOL, and MOL, so these ones here. And so I typed in which supertypes I'm interested in. And then by running all of this, I'm then given this table, which shows me my top 2 differentially expressed genes for my 4 different supertypes that I selected. And then this is all sorted by the adjusted p-value. And so we have that up here in this line. It says sort values by adjusted p-value. And you don't have to write any of this part. This is all in the notebook. You just need to go in and make any specific edits, like how many genes you want, or if you want specific groups, or if you want to look at, change the level from supertype to subclass or cluster. Those are the parts that you would edit. Otherwise, everything else is already written in the notebook for you. And so now I have them sorted here by my smallest adjusted p-values. And then using that notebook again, I went in, I just edited this line here. I edited in my genes of interest from that previous table here. Everything below it, it comes with the notebook. It's already typed up for you. You just need to run the different cells and it produces this really nice heat plot here. And so here we can see how specific these differentially expressed genes are for these 4 different supertypes. And if you have any further questions on how to use these notebooks, if you're a bit unfamiliar with using Python or using some of these notebooks, we had an entire webinar last month specifically featuring Chris. Chris Morrison is the one who wrote these notebooks. And so this is on our YouTube channel right now. It's called Programmatic Access to the ABC Atlas. And he does a full hour walkthrough of how to use these different notebooks. All right, so now we can move on to if there are time for any other questions. During this time, I'm also going to launch a very brief poll. And so this poll is anonymous. It should show up on your screen now. And this is just helpful for us as we're planning and designing our webinars on looking at, what's useful, what's, helpful for planning our next events. So this is all anonymous. So please fill out that feedback survey. And yeah, now we have time, a couple more minutes for if anyone has any other questions, you can please submit them in the question box. Thank you for filling out the poll. If anyone hasn't filled that out yet, please do so. It's helpful for us just for planning and organizing. And yeah, again, if there are any questions now, we have some time for that. If there are no further questions, we can also move ahead just to some final wrap-up slides. Okay, well, I'll move ahead to the next couple slides here. So next month for the webinar series, we're actually doing something a bit different. We're going to be having office hours. So we'll have two different office hours for the month of May. And with these office hours, there will be no prepared presentation. It'll just be free time for Q&A and for discussion. And for each office hour, we're going to have two different scientists from two different teams at the Institute. So for May 7th, we'll have a representative from the mouse cell types team, and then we'll also have someone from the human and non-human primate team who are both working on single cell or single nucleus RNA sequencing. And same with the clustering and taxonomy building webinar on May 20th. We'll have someone focusing on cross-species and non-human primate work, and someone else focusing on human work as well. And so to register for those office hours, it's on the same registration link as the rest of this webinar. And we would really appreciate anyone to submit questions in advance, there's a spot for that on the registration form, just so we have some questions prepared ahead of time for those office hours. But again, that'll just be open-ended. There will not be any presentations. And the first webinar or the first office hour on May 7th will very much be more wet lab focused. So if you have questions on, troubleshooting protocols or techniques, the two people who will be running those office hour do a lot of the wet lab work. And then the May 20th one will be a bit more computational focused. Once we have that, single cell or single nucleus data, how do we analyze it and cluster it and then build taxonomies and build cross-species taxonomies with these data sets? All right. And if there are no more questions today, then we can end the webinar a couple minutes early. This webinar is funded through an NIH grant through NINDS. And we would like to acknowledge our funding and also the support from the Allen Institute. And thank you all for joining us today.